Thank you, and thank you for the invitation. It's really a delight to visit here, meet with many of my dear colleagues, and meet new ones as well. So enjoying the day so far. And again, I had sent a couple of different potential topics to Patty, saying, well, your pick. I'm happy to talk about any one of these. And just for background, on my lab and throughout my career, I've mainly been interested in pollutant chemicals, understanding how microbes have adapted, evolved, how communities develop that start to degrade them, whether they're brominated or chlorinated compounds and sediments. We're working also on natural sources of these compounds, such as marine sponges. Worked on selenium and arsenic-respiring bacteria, and recently on the fate of pharmaceuticals and personal care products in aquatic sediments. This is actually a view of the Raritan River in New Brunswick there in the background. But for today, I want to take you to the Arctic, and specifically to the Finnish Arctic. So area here. Now, what's very nice about Finland, Finns are very organized. No, I'm a Finn. But also, things are clearly marked. So I can go up to the Arctic Circle, and I know when I have arrived. And I can go up north, and now I can get into Arctic research on here. So again, aspects of how I ended up on this. And I'm going to, of course, talk about a lot of work that others did, not me, mainly Minna Männistö, a former postdoc of mine in Finland, who's now at the Natural Resources Institute Finland, Lars Gansert, another postdoc also who was based in Finland, and my graduate student Prashida Gadkari, who worked on some of the key experiments, and several collaborators, Lee Kirchhoff, Jana Bromberg, and Maria Tirola, and others as well involved. And my interest in Arctic soils really got started about 15 years ago. I was on sabbatical in Finland for a completely different project, and had the opportunity to start developing essentially what was a bioprospecting project in northern Finland. Can we start to explore Arctic soil sediments, find new microbes with novel enzymes, novel bioactive compounds, interesting activities that could be used in one part on environmental biotechnology and wastewater treatment in cold environments. This is, of course, an issue, is how do you treat waste when actually it's freezing for most of the year. What can we do in bioprospecting for new psychrophiles? Can we get new organisms that might be producing new chemicals of interest, so new enzymes, pharmaceuticals, but also understanding the microbial soil processes for really a sustainable Arctic environment. How do you interface with the natural environment, but also reindeer husbandry, which is a major livelihood in the north, as well as with forestry in these areas. And that's really the area that we then continued on with funding for many years from the Academy of Finland. So I basically have kept sort of a side lab and group in Finland, in addition to the work that I do then at Rutgers. Our study site that we've been going back to now for close to 15 years is here in Kilpisjärvi. That's a site just at the border of Norway, Sweden, and Finland, way north of the Arctic Circle. So the town of Rovaniemi is where the Natural Resources Institute has one of their main laboratories and stations. And from there, it's at about a 6-hour drive up to Kilpisjärvi, where luckily the University of Helsinki has a field station. So we have very good facilities where we can stay, and from there basically hike up to the Malafell. This is basically has been one of our main field sites where we've been studying now microbial processes in the soils. There's major changes, of course, here in temperature. You have cold winter. The average temperature at this site is about -2 degrees Celsius throughout the year. In the winter, again, it'll be -20, -30, -40 at times. In the summer, of course, temperatures are increasing then to, in some cases, actually, Lapland has been one of the warmest regions of Europe in recent years. Again, shifting climates, this is one of the big questions. So going up to 25, 30 degrees Celsius for a number of weeks. But again, so you have major shifts in the temperature, and this question of freezing, thawing conditions is what we've been focusing in on. So just a few pictures of the site. So again, this is late May, early June. So you have spring, summer, July, August. By September, you're into fall. And then for about 8 to 9 months of the year, this is what the place looks like, or actually might be very dark because the sun doesn't even rise for 2 months in the very north. And the question, of course, is one of air temperature, but to us more interesting is what's happening in the soils. And we've been monitoring at this site with temperature loggers, so we know actually what the soil temperature is. And one of the questions we got into is how snow cover is affecting soil temperature, and then what would be that impact on microbial activities. So again, we've looked at windswept and snow accumulating sites that are just a few meters apart. So depending on the topography, snow will accumulate and you have 1 meter or 2 or 3 meters of snow. Under that snow cover throughout winter, soil temperatures stay very, very stable at -1 degree, maybe -2 at times. A few meters away, where the wind blows away snow and you have just a couple of centimeters, you have major zigzag temperatures going from -5 to -20, -25, again, depending, of course, on the winter. But you see that difference. And so we've been interested then at this site in winter, here in summer, to see what's happening now with carbon-nitrogen processes in these. Now, why we're interested in this, of course, the site, which is really one model site for broader questions, is what's happening to stored organic carbon in tundra as well as boreal soils. They hold approximately 1/3 of the global carbon pool. I mean, the soils that we're studying, I mean, the top soil is essentially 90% plus organic material. And the question is, as these soils, whether they are seasonally frozen or permafrost, start to warm up, how much of extra carbon is now going to be released by microbial activity into carbon dioxide? And the concern, of course, of this feedback loop that then may occur that we don't understand yet. And again, we have both seasonally frozen soils, but also permafrost that are being turned into wetlands, thermokarsts, and again, the question of remineralization of this organic pool. We're, of course, interested in understanding essentially who's there, but more importantly, who is active and when. And how do changes in the conditions, temperature, nutrients, snow cover, vegetation, affect activity? So what's happening from an initial residential community with changes, who else pops up, who becomes active, how can we start to dissect this? And again, so how do changing Subzero temperatures, other cycles, change the community structure, dynamics, activities? And again, how will those changes in active microorganisms affect the overall carbon turnover processes? So that has really got us to focus into carbon as also nitrogen utilization in seasonally frozen soils. Carbon, of course, for CO2, but nitrogen comes in because these are quite nitrogen-limited soils. I mean, there's all the carbon you can get. And we've found that some of the microbes start to degrade the organic matter more as a source of scavenging nitrogen than actually for going after the carbon. So nitrogen plays into it. And a lot of the work, including what we've been doing for many years, is focused really on the short summer period. It's easier to go sample. And again, the assumption is that, well, microbes are active then. And there's been much, much less work done on what's happening during most of the year. It's about 9 months at our site where the soil is frozen. And so who is active in the subzero, under these subzero conditions? And again, so that's where we're really focusing in right now and where I want to go into what we're starting to learn about this system. Over the years, we've taken a number of approaches to just characterize the microbial community. Of course, I'm a classically trained microbiologist. I like to cultivate organisms. I understand that what grows on a Petri plate is not their natural habitat. But what we can learn from these organisms as models and going back to the environment is tremendously important. A whole range of molecular community analysis, and the slides dates how we've transitioned through different methods from TRFLP to now different sequencing technologies, also PLFA profile analysis. And then, of course, using different microcosms and different types of fields experiments where we can look at how different changes are affecting. So I'll get into some of these approaches. Now, one of the early discoveries in these soils was that a new phylum popped up that at the time was not very well studied at all, the acidobacteria. Now they're well understood to be distributed in soils around the world, and especially in acidic soils. The soils at our site are about between pH 4 and 6. Again, it's the humic material that is driving the pH down. And we can see that even within the site, dependent on pH, if you go to a neutral soil, actually actinobacteria, proteobacteria are dominant, go a few meters and the pH drops to 5, the acidobacteria become dominant. Often being up to 50% of the ribosomal RNA gene reads that we can see. So really dominant players. And at the time we started this, there were 3 isolates in cultivation, 1 species described. Now there's been up to, I think, maybe up to 20, almost 30. But if you compare that to the proteobacteria with 3,000 plus described species, we know virtually nothing about this phylum. And again, at these sites, this is clone library data. Again, you can see that there's actually diverse groups within this phylum as well. Now, man is to aim to get these into cultivation. And since we were interested in carbon turnover, we tried a whole range of different carbon substrates, what we could get off the shelf, to just simple R2A agar that we modified with pH, to making moss extract, soil extract, lichen extracts, and so on. Tried all of this, cultivated at different temperatures, 5 degrees, 15 degrees. We haven't yet done sub-zero cultivations, but I have some ideas on that. And then waited and waited, sometimes for many months, to look at some pinpoint little colony come out. And with that, then eventually, Minna was able to isolate the first acidobacteria from our site, which we then named Pteroglobus sanensis. That's after Mount Sana, one of the, actually a very worn down mountain at the site. And again, in the subdivision 1 acidobacteria. Once we figured out how to grow them, we could isolate more and more of them. So The isolates at our site, and this actually coincides with the molecular data, is that they're falling into two now expanding genera in the subdivision 1 acidobacteria, Pteroglobus and Granulicella. Again, the acidobacteria is very diverse. I think based on 16S RNA grouping, there are 26 different phylogenetic subdivisions, of which they're cultivated strains from 5 of them. And just about most of them are coming from subdivision 1. So there's something in here that just these strains like to better grow in the laboratory than the others. We've figured out the conditions a bit more. And we're bringing even more of this. This is just the most recent set of strains from this site where we can see that, yes, we actually are getting a good representation of the diversity of this group also in culture. And that is, of course, useful then when it's fairly easy to get a genome sequenced and get the blueprint of that organism. And so a few years ago, we had 3 of our strains sequenced. We're now working on actually getting the rest of these through the pipeline. And we're able to start to look at what is it that they are potentially capable of. Now, in addition, of course, we had also a lot of phenotypic data. We've been able to try them out on a whole range of different carbon sources and so on. And so the genome data actually just reinforced what we already know. They have a sweet tooth. They like sugars and, of course, also other polysaccharides. And their genome is actually very full of a whole range of different genes involved then in sugar metabolism, transport, and so on. So the so-called carbohydrate active family, so Casey enzymes in there. And actually compared to, at the time, the only 3 other acidobacteria genomes that were available, these were had much higher abundance of these Casey genes. Again, fits into what we think is really involved. They have a heterotrophic saprophytic lifestyle in these organic-rich soils, feeding off partly on organic carbon coming from plant litter, but maybe also some ideas that they may be eating organic carbon slimes produced by other bacteria, so extra polysaccharides. So this is where we're thinking of this. The other interesting aspect about the acidobacteria is that they have peculiar hopanoid lipids in their membranes. And of course, from the genome, we could find all the genes involved in hopanoid synthesis. And these are thought to play a role in, just like sterols in eukaryotes, in membrane stability. Now, is this a way to survive heat stress or cold stress? We don't know yet. And so we've tried various experiments to basically cultivate our strains at different temperatures to see, okay, when are they upregulating these? And a lot of really nice experiments that, unfortunately, the data portion of it failed, so we had too much variation. So I really don't know right now what is driving it. But again, based upon other knowledge we know about hopanoids, most likely this plays a role in terms of how they can survive this habitat. But let me really get to the main part of the story. This was really just background on the site into figuring out what's happening under Subzero conditions. And we've taken a number of different approaches on this. One is using stable isotope probing as a method to see who is assimilating carbon and synthesizing new genomes out of this carbon. Can we determine who's active in situ by actually measuring ribosomal RNA? Again, the idea is, well, if you're metabolically active and making proteins, you need ribosomes. And that's been shown that the ribosome count of an inactive bacterium drops to low levels, and the moment they start growing, get upregulated. So again, comparing DNA, RNA profiles on this. And of course, then a whole range of different microcosms where we're feeding different litter or other carbon substrates, again, to see, okay, who turns on when and under which conditions. Prashida Gadkari graduated based on this work last summer, working on the SIP work with the site from Malta. And again, I'm going to focus mainly on our work of using cellobios. We also tried other carbon sources, also nitrogen sources, but we don't have the data from these systems yet. And again, the basic experiment, taking a soil sample from the site to the laboratory, setting up small microcosms of about 0.5 gram in size, feeding them either C13 or C12 cellobios. I'll come back to the reason why this is critical, and then incubating them at 0, -4, -16 degrees, and waiting sometimes up to 40 weeks until we take a sample or sacrifice that whole microcosm, do DNA extraction, then cleanup and separation in a cesium chloride gradient, where you can now separate your C12, C13 DNA, and then use those two fractions for our molecular analysis, eventually into Illumina MySeq analysis on there. So the idea, again, essentially is one of adding substrates, extracting DNA, separating, and then different PCR steps then to look at the amplicon data. And the question again is, if you have your experimental substrate, okay, you will see the organisms that are using that substrate in your C13 band. If you feed C12 substrate and you take your heavy DNA, you'll see nothing. And that becomes critical in our control on here, and just showing the data from our sample sets. Now, this is work that we're doing together with Lee Kirchhoff at Rutgers, who's really been pioneering the design of this approach. And one of the additions to the SIP protocol that he has introduced is to add a C13 DNA carrier to your sample before you do your separation. I mean, the reason is that you get very, very little incorporation. So if we just relied on what cellobios got incorporated into new genomes, we wouldn't see that band at all. Even though, so here we're adding now archaeal DNA, and then when we're using bacterial primers, doesn't matter. So we're just using this to visualize and find that band. We can pull that top band and that bottom band, then analyze the bottom band, and again, in our C12 controls, it's flatlined. Again, if you run your PCR long enough, of course, you're always going to get a signal from nothing. So basically, the important aspect here also is knowing, okay, how many cycles can we go and ensure that we still have our control flatlined. And again, when we pull the heavy band from our C13 fed samples, you now see a signal. And we're using TRFLP initially as a very quick screening tool before we then decide, okay, we're then taking out for Illumina sequencing. And this shows just that same data set from one of our replicate cultures. Again, our C13 fed cellobios, so the bottom, the heavy DNA band in red, and our control completely flatlined. And so this, of course, tells us there's a whole bunch of different cellobios utilizing members in the community, even at -16 degrees. Took 40 weeks to get there. We tried earlier time points, 5 weeks, 20 weeks, nothing. And so we think that we're basically just at the detection limit of SIP, meaning you need at least 2 genome replications in order for you to have one fully labeled C13 heavy DNA strand to separate. Again, whether they might have gone to 2 or 3 replications by that is probably closer to it. I don't think we'll detect that early one. But again, an example, and we have triplicate independent microcosms analyzed separately, all showing the same data. I'm starting to believe this. Initially, I wasn't sure on that. And again, looking at then the data from our Illumina set, here is our stat bar graph of the, sorry, I had to add one of these. Initial set, basically, we have number of different proteobacteria, actinobacteria, acidobacteria. And as you can see then, after 5 weeks of incubation on cellobios at 0 degrees, -4 degrees, or 40 weeks at -16, major shifts, new groups coming up that were there, but not as abundant in the community. Again, Candidatus saccharibacteria, the Ignavibacteriaceae, the acidobacteria that we thought would, we know that already that during the summer they grow slowly and are outcompeted by proteobacteria. That, maybe they get the upper hand under Subzero conditions because they can just chug along and use all that cellobios. No, they essentially disappeared almost completely out of this. They're not that active at Subzero temperatures. We see, again, that same data set, then looking at the principal component analysis, again showing that, yes, incubation on cellobios, well, one, it's of course looking at a subset of the community that can use this specific carbon source. That would be different, but also, again, our triplicate microcosms at the different temperatures are separating, especially the -16 separated from -4 and a -0. So again, giving some belief that, yes, these are true activities on here. And we can look, of course, down also with Illumina data down at the family level, where, again, what are particularly interested are the Ignavibacteriaceae, and especially a family called the Melioribacteriaceae, coming up very much. It's barely present. I hadn't even looked at this organism before or paid any attention to it. Actually, not much even heard of it before. I've heard of the saccharibacteria. They're coming up before. And again, they're detectable in the initial community, but again, clearly coming up. So those are a couple of groups that we looked at more that were clearly shifting during this Subzero incubation. And just a third way of looking at that same data set, a cold map, as my student called it, where, again, you have your initial community that is not active on there, and again, completely new players that are now utilizing, assimilating cellobios at these Subzero temperatures. Which sort of makes sense, yes? Microbes will have different temperature optima, and who's active at 20 degrees in the summer is probably not going to be utilizing cellobios at -16, -20 degrees in the winter, and so on. And so understanding these different temperature optima becomes very interesting as well. The other factor, of course, is, well, what do you need in order to be active at Subzero temperatures? Liquid water. That means in order for them to be active at -4, -16, they must be living in some kind of a brine pocket that's still left. And is my idea of how I'm going to cultivate them is go for osmophiles, halophiles on the system. And there are actually, there's one strain from Lyle White's group in, from McGill that grows to -13 degrees that they've been able to pull out from one of their sites. So yes, this is the question, is how can we now, we know they're there, we know they're active, is there a way that we could start to selectively pull them out? So again, variations on who is dominant initially versus active at 0, -4, -16 degrees, and again, Saccharibacteria, Melioribacteriaceae come up. And so with our data, we have enough sequence length to do at least some phylogeny on there. And there actually are 2 cultivated species of the phylum Ignavibacteriae. Ignavibacterium album and Melioribacterium rosius, both coming from hydrothermal sites. And actually all the other 16S RNA sequences in the database are coming from a hot spring clone, an olefin clone, a hot biofilm clone, volcanic lake water clone, mainly thermal environments. What are we doing now with cold active bacteria in this group where basically nobody's seen them before in this habitat? Not what we were expecting, definitely not. So it's fun when the unexpected happens on there. So what we think is, well, we've now shown that, well, the Ignavibacteria are not just thermal, found at thermal sites, but actually there now is this new Subzero active clade that we need to explore more. With the Saccharibacteria again, here our data fits in what's been shown from a number of other sites. So our OTUs here in bright blue, but again, other clones closely related from Arctic, Antarctic sites, and the same also with the Vericomicrobia that came up again, found diversely in a number of Arctic, Antarctic, as well as alpine sites as well. So again, finding organisms active that we really weren't prepared for, and that's changing our thinking now. We still have no clue of what makes the acidobacteria so abundant. That's going to be another story on there. But also showing that no, this works also in other systems. This is from an earlier paper with Lee Kirchhoff's group, where we showed that the CYP protocol worked with permafrost. And again, taking a permafrost sample that had been frozen, I think, for 80,000 years or so, without thawing it or quickly adding, in this case, acetate, incubating them for many, many months. Again, finding who's eating acetate. These are just the different OTU groups. But what we're seeing is that one group was active at 0, -3, maybe -6. Then we have another group that we only found at -6 to -9 degree incubations, another that were only in the really, really cold incubations down to -20, and then others that were eating acetate under all temperatures. And then others that were sort of here and there, that could either be, this was still using TRFLP method, so it could be the same peak is actually represented by 2 different organisms. But here, this was very interesting indeed, that yes, the Subzero active bacteria have distinct Subzero thermal optima as well, which is of course exactly what we see at temperatures above 0 of different psychrophiles, mesophiles, thermophiles as well. And so that's one aspect that also to think about, well, what's happening with permafrost? Even if it doesn't completely thaw, it's warming. So a permafrost that before was at -10 degrees and due to change in climate is now -4, -2, well, you're going to have different members active. And the question is, will this change what carbon is being turned over? Or is there just so much functional redundancy that it doesn't really matter who's active, they're all doing the same thing anyway, which is another way to look at microbial ecology. Why figure out who's there? Because there's so much redundancy in the activities. Other than that, it's fun. Well, the other aspect that we wanted to do is go back to our site in winter and not in summer. When I suggested this to Minna, she said, well, you better show up for that sampling trip then, which I did. I was taking the pictures while they were working. So this was in February, so it's already getting a bit warmer, a little lighter. We have at least 5 hours of daylight, so we can get up, leave 8 in the morning to our site, hike through the darkness up, which takes, well, with snowshoes, about an hour and a half to our site where we had actually temperature loggers that were there from the summer. And again, we can find the exact same locations that we've been sampling now for many years in the summer. Dig down into either the snow accumulating site. This is the technique of Lars working on this. Again, ended up being about 2 meters of snow. And then while he was digging one new plot, Minna was in the other, sampling with our high-tech sampling equipment, a hammer and a chisel, to basically break off enough soil that we can put in a pouch and bring back then to the lab that same evening. So we spent a day doing this. It's actually very nice and warm down there. It was windy and actually not too cold, only minus 10 degrees up on there, but with the wind chill, it's worse. So getting down below the snow is actually a good way. And so again, we have our soils, we bring that back. And then just a few meters away is our windswept site. So it had snowed actually the week before, so there was about 5 centimeters of snow, brushed that away, and then use our same technique to do, and I actually helped a little bit with that part as well. Bring those samples back to the lab. We can now extract DNA and RNA. Minna has developed a really good CTAB protocol that gets actually good quality DNA and RNA from these high humic soils. And again, this is still using TRFLP methodology. We're still working on our ion torrent sequence data from the same set. Again, showing that the windswept snow accumulating site, many of the same members are there, but there are also clearly distinct differences, and also differences between who's there resident in terms of DNA versus who's active that we can find in RNA. And that shows very nicely with this MDS plot of that data set, showing again our DNA set clearly separated from the RNA. So again, who is active, having ribosomes at that time in February is a different subpopulation of the community. And also we're seeing differences between also the snow accumulating, and so in S, and the windswept with a W also. So there are differences also there in terms of the temperature optimum. So we're now digging into this data set to be able to compare also with our SIP data on who's there and active. Again, the SIP data, of course, is selective because we've used at this point only one carbon source, cellobiose. So here, of course, there's many other things to be eating, so that becomes more complicated. Most recently with Lee, we're looking at using the Oxford Nanopore MinION sequencing data set. So basically this is really coming from Lee Kirchhoff's lab, who's been running with this new approach and liking it very much. One, because you can buy a sequencer for $1,000, and then a flow cell is another $1,000, so you don't have the huge initial capital expense. But more importantly, you get long read lengths. So my issue as an ecologist looking at short Illumina reads is I can't identify beyond getting to the family, maybe genus. You only have to go to look at the proteobacteria to see how much divergent physiology there is even with closely related. I have a few minutes left? Okay, I can get in. So that was one. We had data sites again for our deep snow windswept site. They separate, they work. We focused in here on the acidobacteria to do one step further. Now, the problem with the sequencer is that it has a high error rate. And it's 5%, 10%, it's going down. But that means it's a random error. And so what we need to do is bin the groups of sequence raw reads that we think go together. Then we can align them and build a consensus sequence. And actually this shows here is that we're now being able to do phylogenetic analysis using the full RNA operon, so it's about 4,500 base pairs of data, which gives us even strain level differentiation. And so that is what I want to know. Okay, things that we now just lump together, maybe they are shifting under different conditions, not only in the Arctic, but of course other sites where we're looking at that same aspect. And of course, we also have ongoing things with adding different types of litter, again showing Impetrum vaccinum. These are the main small understory brushes here, and then incubating at different temperatures. This is 5 degrees minus or +2, -1, -5. Again, we're now going through the communities and see who's eating this natural litter coming in and linking that to our SIP experiments as well. So we're learning more and more, and we're learning more and more things that we still don't understand and know and need to start to explore. Again, there are, not unexpectedly, Subzero active microorganisms at this site that is frozen for most of the year. Again, diverse community of not only the Ignavibacteriaceae, the Saccharibacteria, but also other Proteobacteria, Plactomycetes that are clearly eating cellobios at Subzero temperatures. Again, the Acidobacteria, Actinobacteria that are really dominant in these soils did not really show up. So we still need to understand what is it that makes Acidobacteria so dominant, even though they're slow growers, don't seem to respond to anything. You add anything else to the soil and Proteobacteria bloom. Acidobacteria just chug along. Maybe they don't die, and that might be the aspect. So we need to think at mortality more. Again, divergent lifestyle is going to be reflected in different usage of different carbon sources. That, of course, we need to be able to now to start to integrate with climate change models and so on to understand what is the consequence of changing climate to this carbon loop. And so linking all of that together and so on. The other aspect, of course, it affects plant growth. And so Arctic greening, and that effect is now an aspect that Minna and Sari Stark are working on and continuing on this as well. So that adds the effect of plants onto the system. Again, one can do simple experiments like this that increase the soil temperature by 2 degrees on there and be able to model that over many, many years. Reindeer grazing adds one more element to that, and so on. I'm going to jump over that, and I want to do one final plug for something completely different, except it is an organism that we do find here. The New Jersey state microbe, Streptomyces griseus. I can now announce that it's basically at the final step waiting for the governor to sign, and he has until May 10th to do this, and if he doesn't, it becomes law anyway. It's passed the Senate, it's passed the Assembly. So Streptomyces griseus, the producer of streptomycin, the first cure for tuberculosis, is now going to be the formal state microbe of New Jersey. It's, of course, also the organism that gives the wonderful earthy smell and produces a whole bunch of other compounds. But that's been a long, multi-year initiative, lots of lobbying and so on. But hope Alabama is next.
We got time, I think, for a question or two.
So you were talking about some of the active microbes are similar to the microbes that live in the hot spring, extremely at pretty high temperature.
They're in the same phylum.
Okay.
Which means they could do clearly do completely different things. This is the problem of lumping everything together. Oh, everything we know is a thermal organism, so these must be there as well.
Okay, but so my question is, because I'm not a microbiologist, right? There are so many species, genres are there. But you were also mentioning from the more like biochemical perspective, that when you look at some hopnoid that actually regulate the memory stability. I find that's very interesting. So I'm wondering, those similarity, which means those bacteria are pretty stable in tough conditions. Have you looked at if they share similar hopnoids? And
the others don't, at least from the two isolates that are known. No, they do not have hopnoids. This is a peculiarity of just a couple of bacterial phyla. And why the acetobacteria have them, we really don't know. But no, the other ones here that were active, they do not, as far as any of the data that I've seen from cultivated representatives, have hopnoids. I mean, our goal is, of course, if we really get the CYP to work, is to be able to now do a metagenomic sequencing and be able to put their genomes together even without having cultivated them. And so that is going to be, it's in my proposal right now. I'm waiting to hear back from actually the Academy of Finland and then need to resubmit to NSF once again for this. It's a great idea, but they still don't want to fund it.
So when you were doing your metagenomic sequencing, are you finding a lot of lateral or horizontal gene transfer evidence or any type of extra chromosomal elements? I'm just wondering whether
in the acetobacteria, no. And those are the ones we focused. We right now are working, so this is Barney Whitman's program on the pangenome, because we have 1,000 strains from the site. Not just the acetobacteria, but a whole range of proteobacteria. So we're actually growing them right now to send for sequencing, where we now have either within the same species or very close related species, many independent strains. And so hopefully that's going to give some of that data, but you're going to have to wait for an answer at least 3-4 years until we get the data back. Actually, the difficulty is not sequencing, it's growing the strains to get enough DNA. The sequencing goes overnight.
All right. Well, let's thank Max and
Thank you.